to this lesson for Advanced Higher Biology. We are in Key Area 1 of Unit 2, and this lesson will be looking at how we can monitor different species populations. Prior to now, we have looked at the identification and taxonomy of species. We're going to focus now on how we monitor those populations before moving on to looking at measuring and recording animal behaviour. So for this lesson, we're looking for you to understand the reasons behind and the methods of monitoring populations. You need to be able to explain the need to monitor population numbers, describe the role that is played by indicator species, describe the process of mark and recapture and when its use would be appropriate, and give the assumptions made during mark and recapture studies. Finally, you should be able to list the methods used to mark organisms. So when we look at population monitoring, this usually involves counting or estimating the number or the density of a species on more than one occasion. So we don't want to simply provide a one-off inventory of the species in an area. Instead, biologists are really aiming to detect changes in populations over time. And by doing so, they can determine any natural phenomena that might be occurring, any human interactions that might be occurring, and if they are taking steps, any management of that species, they can look to see if that's responsible for any changes. So this graph is looking at the different population changes in bird species in Canada, and as you can see, since 1970, several of them have declined, several of them have increased, and biologists will be looking to investigate the reasons for that. Now, of course, there are some species whose population numbers can actually suggest specific changes in the environment. So rather than looking to investigate why a species has changed, the actual presence, absence or abundance of that particular species will tell us something about that environment. And these species are of course referred to as indicator species, which is an idea you should already be fairly familiar with. So these are the species which are able to provide information about environmental conditions. Indicator species are particularly useful when we're looking for the presence or absence of a specific pollutant. Now, all organisms have a preferred niche, the preferred conditions that they are able to survive in, and they also have some which they are intolerant to, which essentially makes the niche unavailable to them. Where we have a specific pollutant, we might have the presence, absence or abundance of a species which is either intolerant to that particular pollutant or favoured by that particular pollutant. So it might be that that pollutant provides conditions which are in the organism's preferred niche, so therefore it is a favoured organism. The presence of that organism therefore indicates the presence of the pollutant. Equally, we may have some which are intolerant to that pollutant. That would make their conditions to be in their zone of intolerance. Consequently, the absence of that particular species suggests the presence of that pollutant. And we can see this example when we look at lichen as indicators. So different forms of lichen have different susceptibilities to sulfur dioxide pollution. Hairy lichens are very susceptible to sulfur dioxide, so when they are present, that indicates a low sulfur dioxide level. Shrubby lichens indicate a medium level of sulfur dioxide, and leafy lichens indicate a high sulfur dioxide level. So if we only have the more tolerant form, the leafy lichens, if they're found in an area, then we can say that the area is experiencing relatively high levels of pollution. Whereas if we have the most susceptible forms, or hairy lichens, then there must be very little sulfur dioxide pollution in the environment. And if we're regularly monitoring our lichen populations, that can provide additional data about our changing air quality. Now, as we discussed in a previous lesson, it is impossible to study every member of a population, and similarly it would be very difficult to count every member of a population. And we considered the fact that we can use different sampling techniques in order to look for a representative sample of the population. Now, with some species, it is not always possible to recognise specific individuals, and that means they could be counted more than once. So we need to consider techniques that we can use to avoid that particular issue. And this is where our mark and recapture technique comes into play. So this is a method for estimating population size. A sample of the population is captured and then marked and released back into the general population. This marking technique must obviously have a minimal impact on the species being studied, so it doesn't interfere with the individual's normal behaviour or indeed make it more conspicuous to predators. But it must be clear enough that you can see it again. Now, after an interval of time, a second sample is going to be captured, and we're looking to see if any of those individuals are recaptures, so if they've got the markings from our initially captured population, and that allows us to make an estimate of the total population. And we do this by carrying out the following equation, where n equals an estimate of our total population, m is the number captured, marked and released in the first sample, c is the number captured in the second sample, and r is the number of marked recaptures in the second sample. Now, we do make several assumptions when we are using that particular equation. First of all, we are assuming that all individuals have an equal chance of capture, that there's no immigration or emigration during the sample time, and that the sampling methods used each time are identical. Now, our methods of marking are going to vary depending on the type of species that we're going to use. First of all, we have banding, and this is where bands which have got an individual identifier are going to be attached to the animal, and ideally this is going to be placed where it's not going to impede their movement. Depending on the observations being done and the species going to be used, it might not be possible or appropriate to physically recapture our organisms, however. So in the case of monitoring migration patterns, or where organisms might be cause significant distress if they are subsequently recaptured. In those cases, it's possible to use different coloured bands or sequences of coloured bands, and that can be used during remote identification, such as when you're doing a point count or using binoculars, for example. We can also use tagging methods, and again, these are going to vary according to the species that you're tracking and the method of sampling. So some tags, like the one on the left on our turtle here, can be used for remote tracking using radio transmitters. And consequently, the tag needs to be securely attached, but again, not interfering with the animal's normal behaviour. It is also possible to have some tags which are surgically implanted, and that prevents them from interfering with the animal. So for example, external tags on a fish would make quite a difference to its streamlining capabilities, and consequently a surgical implantation is better in those cases. And obviously many pets have been given microchips as a means of identification should they become lost or stolen. We also have paint marking, and just like it would suggest, this is where a code or a mark is painted onto the surface of the organism. Obviously this needs to be a non-toxic and biodegradable paint. We also need to make sure that the paint does not block any skin surfaces that are involved in heat regulation or any secretions or indeed respiration, as some organisms are able to transfer oxygen through their skin. It's also important to consider the impact of scent marking on animals and any conspicuous markings that may make these animals more obvious to predators. Lastly, we have hair clipping methods, and this is where a clearly identifiable section of hair is removed from an organism, and that can be used as our method of identification. Obviously, we need to take care to consider the size of the clipping, any distress caused to the animal, and if those animals indeed use hair or fur in any kind of social signals or in any kind of mating rituals. So from that, you should be able to explain the need to monitor population numbers, describe the role that indicator species play, explain the process of mark and recapture and when you might need to use it, give the assumptions made during mark and recapture studies, and list the methods used to mark organisms. As ever, please do get in touch if you have any questions. Either leave a comment, visit the website, or send me an email on the address that's to follow.